Okay. So put your name in the pocket. First unit we're going to go over is organic molecules. Now, the kids kind of showed me that the board when they opened up this document is kind of sometimes like not fitting the words in the right places and whatnot. So sorry that these pictures are off, but you're pretty much going to get everything that I have. Okay. So, first thing we're going to talk about are the 4 biomolecules. Remember, carbs, lipid, protein, nucleic acid, chon, chomp. And the enzyme unit goes with it because enzymes are a protein, okay? So, first thing I'm going to do, and if you ever have a biomolecule question, think of the order that we learned them. Carb, sugars, turns into fats.
Hey, I'll call you after a while.
Okay.
I just need a favor.
Okay.
But not today, like in a week.
Hey, that's fine. Proteins, and then nucleic acids. So, with the elements that go with them, I'm going to right away write CHO, lipids are CHO, proteins are CHON, and then nucleic acids are CHONP, CHONP. Okay, so carbs. You know about carbs, it's just energy. So, no matter if the body uses it or cells, and we don't really have enough time to go over all of this stuff, so I'm going to give you the highlights. The body and the cell uses it for quick energy. And think of carbs you eat for quick energy. They're kind of light. They're not that dense. We'll talk about that in a second. But they're quick energy. The monomer, one carb is called glucose. Monomer is 1 unit, and then polymer is just when you have a bunch of these kind of together. The polymer is starch. And then the carbs also can be called saccharides, so you can have a monosaccharide or a polysaccharide. SACCHARIDE. That's in one question on here. So in case it says, what is the monosaccharide or the polysaccharide? I always think of sack of sugar. So it's a sugar. Okay. Now this picture, if I would count it out, there's 1, 2, 3, 4, 5, 6 C, so it's C6, H12, O6. All carbs, no matter what, are in that kind of ratio where you have double the hydrogens. It could be C12, H24, O12. It could be anything as long as the H is doubled. Okay. You eat too much sugar, you get fat. So lipids, the slang name is just fat. Okay. Now back to what I was saying where carbs are usually light and kind of airy. Fats are kind of like compact and dense. What are some fats you could think of? Like fat on meat, like it's very dense. Oil, okay, lard.
Oh, yeah.
Okay, so if I would get you, some lard in a container versus some like Rice Krispies, the carby Rice Krispies are very light and airy, not as dense, versus the lipids, which are very dense. And so, on a gram-to-gram ratio, who has more energy? Is it the lighter or the more dense?
Lighter.
The more dense.
Oh, yeah.
Okay. Think about it. If you were starving, would you want a bowl of cereal or a bowl of like lard? I know nobody wants to eat lard, but that would give us more calories, okay? So lipids are fats, and they're used for energy storage, storing energy. You store energy in the fat in your body. So we're going to say this is quick energy, this is stored energy. Okay. Now, one lipid is called a fatty acid glycerol. I'm just going to write a FAG to stand for fatty acid glycerol. STAR test really doesn't ask about that too much, but they do ask about the polymer, which are fats, oils, And so we need lipids for like insulation. We need lipids to surround our cell membrane. Remember the lipid membrane is made up of fat. Okay. And now, even though carbs and lipids are both CHO, I'm gonna come up here to the carb one and put a 2 next to the H. It will always be in the 1 to 2 to 1 ratio versus lipids are random. So guys, if I would count this out, this right here is like C30, H75O6. That is in no ratio. So if you would ever have to identify a lipid, it would kind of look like a carb. Like it could, this could almost look like that, but there's not enough O's to be in that ratio. It would have to be like C30, H60, O30. You know, it'd always have to be double the H's. Okay, next one, proteins. Proteins, CHO. So this is where you have a nitrogen, here's a nitrogen, but you don't have the phosphorus, okay? And the function of a protein is to build. And I lie, it's not just building, because let's look at enzymes. What we're gonna do is we're gonna take a break and we're gonna do the enzyme part, because enzymes, just like this Pac-Man that just eats the dots, eats the dots, eats the dots, every time it eats a dot, does it take a rest? It just keeps on going, keeps on going. It's the Energizer Bunny. So enzymes, they don't ever run out of energy. They just keep on going, okay? Now, if you look at this picture right here, the purple's the enzyme because it just keeps on going, keeps on going. Okay, the purple is gonna attach to the green. It's gonna break it in half, okay? And it's just gonna keep on going, finding another green, breaking that. Now, this green molecule would break on its own. So guys, if you are not producing enzymes, you will eventually break whatever you need to, but you're gonna have to wait a long time and you're gonna have to use your energy. Guess what enzymes do? They speed up the reaction because they use their energy. So we want enzymes because we don't have to use as much energy. So people who can't lose weight, guess what they're probably lacking? Enzymes. So guess what a lot of weight loss pills are? Yeah. So what biomolecule are they? They are pea proteins. So fill that out. Now what do they do? So this is the thing. We're gonna say break or build. Break or build using their energy. Now, technically, what the textbook definition says is it lowers the activation energy. Basically, if this thing has to break, it gives its energy to it and lowers the energy it needs so it breaks on its own. Now, this might be fat being broken down, but guess what? How do we build muscle? We take 2 amino acids, we build them together. So protein or enzymes, they can break when it comes to like breaking down fat, or technically they could build fat, or they could build protein, or they could even break down protein, okay? It always ends in ACE, luciferase, glucase, lipase. Remember we did the lab toothpick ACE, okay? Enzymes end in ACE. What kind of conditions are optimal for enzymes? This is what we're gonna put. We're gonna put neutral. Picture enzymes like you guys. Do you like it too hot? Do you like it too cold? Neither do enzymes. Remember when we put our hand in the freezing water and it kind of cramped a little bit?
No.
That's what happens to enzymes. So when it comes to like really high pHs, really low pHs, really high temperatures, really low temperatures, enzymes don't like that. They like being in the neutral, okay? When they are in super high or low temperatures or pHs, they do something called denature. If I break down the word, D means not, nature means natural, so it just means to fall apart or break down. If you want to denature an enzyme, you put it in something hot. You boil it. So guys, if you're eating raw vegetables, you are getting a lot of really good enzymes from those vegetables. What happens when you steam them or boil them like in a soup? Yeah, you're breaking down the enzymes and they don't work anymore. So the reason why it's always so much healthier to eat raw food is because they still have working enzymes in them. Even if you buy them from the grocery food store and are like, how do they still have enzymes in them? If that food is not spoiled and that food is still like good, it's got good nutrients in it, good enzymes in it. So it's always better to eat raw versus cooked. But, I mean.
What about meat?
Okay, so technically raw diets are very good for you. Now, only certain things can be eaten raw, but think about like animals in the food chain. Do animals cook their food before they? No, and they survive fine. And animals are way healthier than humans. So technically, raw food is really not that bad. Okay, here's the picture. What I want to remind you is it's the enzyme that doesn't change. Enzymes don't change. The substrate changes. Okay, now remember, enzymes are a lock and key fit. So if the enzyme looked like this is crazy looking, sorry. Okay, the substrate needs to look like this. Okay, it's a lock and key fit and they're specific for each other. Okay, what happens is when this lowers, it touches this activation site and triggers it to do the breaking or the building. Okay, so ready? You got an enzyme, fits exactly with the substrate. They're touching at this thing called the activation site. And then that triggers the reaction. Now you have a new substrate, but you got the same old enzyme. Okay? Okay, that is a protein. So going back to proteins, there's nitrogen in them. They build or break. And real quickly, the monomer is an amino acid. And the polymers are protein. Or, you know, on a bigger scale, maybe like muscle. Okay? Okay, last but not least, nucleic acid. So remember, DNA, the D comes right here, DNA, deoxyribose nucleic acid, okay? So when we talk about DNA and RNA, those are nucleic acids. Chomp, we got the nitrogenous bases, and we got the phosphates with the phosphorus, okay? How do our body use it? Well, I think our DNA gives us our identity. Our DNA is our genotype, and kind of like who we become, who we physically look like is our phenotype. So nucleic acids give us that genotype and phenotype. The only thing I want to mention about here is DNA is the polymer, and all of these, sugar, phosphate, base, this right here is a monomer, which is a nucleotide. Nucleotide. Okay, last but not least, before I let y'all go and answer those STAR questions, I think there's only 10 or 12 of them. You're going to read them yourself, and then we're going to go over them. Okay, tell me which biomolecule this is based on the elements. Okay, this is CHON. Because I see carbon, hydrogen, oxygen, nitrogen, but no phosphate.
Proteins.
Good. This one is, I see carbon, hydrogen, oxygen, nitrogen, phosphorus.
Nucleic acid.
Nucleic acid.
Okay, these two are both CHO, but when I count it out, this one is C6H12O6. This one is C7H13O2.
Proteins, carbohydrates.
And good, what's the second one?
Lipids.
Lipids. Good. Okay, so I'm going to give you 10 minutes, then we're going to come back, answer those, and we're going to move on to the next unit, which is cells.
So we answer all of these.